Good evening, this is Miss Dagenford. We're going to start our tour of the cell, and we'll be continuing our cell unit by looking at the nucleus and the parts of the nucleus. Now, this is going to continue on with other structures that compose something called the endomembrane system, and this is a part of the cell that is a grand assembly line, but we're going to first look at the nucleus. After this video and the video following this, you should be able to identify the organelles in the endomembrane system, and then identify organelles throughout the cell, but then also be able to explain the purpose of the endomembrane system. This is going to be really applicable in a lot of the organisms that we look at, and you'll see that we're going to give some real good examples of this. So let's get started. So we start with a nice generalized picture of an animal cell. Now, very few cells look like this. Here you have a cell that has everything in equal proportions and has a flagella and then microvilli, and very few cells kind of have this structure. And you're going to see throughout this week, we're going to do a microscope lab that does some comparisons, and you're going to see the differences between the cells. Let's identify some of the main ones. Well, we know we have this large purple structure. That's going to be the nucleus. The nucleus is surrounded by the nuclear envelope. And then within the nuclear envelope, you have these little purple, and I can zoom in on that. We have these little purple guys. They're the nuclear pores. We'll go over those in a little bit more detail in just a minute. And then within the nucleus, we have the chromatin. And then this dark area, the nucleolus. Surrounding the nucleus, you have the rough ER. That's going to be in the blue. The rough endoplasmic reticulum. On the endoplasmic reticulum, you have the ribosomes. And hopefully, as I'm going over these things, there is a part of you that might be remembering things from your previous years of biology or anatomy. Over on the left side, we have the smooth ER, so-called because it does not have ribosomes on its surface. You have the Golgi down here, the Golgi apparatus or Golgi body. And this is only showing one, but some cells have multiples of Golgi bodies. That's no problem. These are going to be our little transport vesicles, or sometimes books just call them vesicles. Very small, spherical, membrane-bound, just transport. They transport things. The yellow structures are going to be our microtubules. Some of you might recall a video we watched. We'll watch it again of our little transport molecule being tugged by a motor protein on a microtubule, and we called the video that we watched called him John. So those are our microtubules. Down here we have a peroxisome. We have our mitochondria in our orange. This yellow body is our centrosome. And inside that we have our centrioles. And so the whole thing is the centrosome. These little cylinder things, those are the centrioles. And then up here we have a lysosome. And you saw the flagella, the long part, and then down here we have our microvilli. There are going to be more things that we're going to look at, but that just gives you a nice tour. So let me zoom back out, and then we'll continue on. So this is just a nice electron microscope picture. These are some simple columnar cells. in an ovary, and here are little microvilli on the top. Your book outlines one, and I'm just outlining another one. Here's our nuclei down at the bottom. And so again, you can kind of get the impression that, each cell has its own unique shape depending on what it does. Now we're going to start something. We're going to finish this in another video, but we're going to start looking at something called the endomembrane system. And we're going to start by looking at the nucleus. Now in eukaryotic cells, the nucleus is going to be really important. It's what can help us understand the genetics. It's going to hold the chromosomes within the eukaryotic cell. So this contains the DNA loosely. In most of the time, the DNA is kind of loosely assembled along chromosomes. In the phases of mitosis, the chromosomes can condense, and you might recall they get really, really, really dark, and you can actually visualize the chromosomes. But most of the time, we just see these loose threads of chromatin. And the chromatin is pictured down here. And the chromatin is made up of the blue, the DNA, and then you'll notice that the DNA is wrapped around these proteins called histone proteins. And these histone proteins are going to be really important later on. We're going to see that can help regulate what genes are turned on and what genes are turned off. But you can see what happens is they get coiled and coiled, and then they coil up some more, and they make these nice threads of the chromosomes. Over on the left here, you can see the nuclear pores. And these pores are made up of these polypeptides. So here's a good example of a big superstructure, a quaternary structure, where these polypeptides are assembled together to make this larger structure, these pores that allow RNA to get out and proteins to come in. What they're showing here is the lamina. This is a network of proteins that creates a supportive network underneath the nuclear envelope, and it just creates a structure to it. And then the other thing we have would be the nucleolus, and the nucleolus is this dark area. And the nucleolus is going to assemble ribosomes. Now, this can be easy to forget, and you get it mixed up with ribosomes. Ribosomes make proteins. The nucleolus assembles the ribosomes. And so what's going to happen is the proteins that are needed to make the ribosomes are going to come in from the cytoplasm, and then the RNA is made by using instructions from the DNA. And so both of these come together in the nucleolus, and the nucleolus assembles these ribosomes, which then go back out to the cytoplasm. And that becomes really important. So any cell that's making a lot of protein is going to need a lot of ribosomes, and then therefore the nucleolus is going to be very big. So that's starting off our endomembrane system. Now, the endomembrane system is going to be this real important assembly line of organelles, and essentially what it's going to be important for is creating the whole population of proteins that cell is going to need. So you want to make the connection that here's the nucleus, and the nucleus, even though it has all of the DNA, not all of it's going to be used. So of all of the DNA that cell might have, only a certain number of genes might be activated. So I'm just kind of highlighting, if this is all the DNA, what I'm highlighting in the red might be all of the genes that you need. And then from those genes are going to be active. So let's say this is a stomach cell where you're going to need a certain number of generic proteins. We call those housekeeping genes, like all cells have them active. But then you're going to have genes that are solely for the stomach functions. And so those genes should be turned on. So what we're going to look at then is how that assembly line differs depending on what kind of protein those cells are making. And it's a really amazing thing to look at. And the more you dive into it, the more marvelous it is. But that gives you a starting point for the nucleus. Really important to kind of examine. Thank you, and we'll continue in the next video.